am Ankita Tiwari, PhD scholar from Department of Pharmaceutical Sciences, Dr. H.S. Gaur University, Sagar, with an interesting and learning episode of B.Pharm on the important unit of steroids 2, which are the very important units of the section of Medicinal Chemistry 2, 6th semester, which is prepared by Dr. Vivek Kumar Vyas, Assistant Professor, and Professor Manjunath Ghate, Professor and Head Department of Pharmaceutical Chemistry, Institute of Pharmacy, Nima University, Ahmedabad, India. Let's start our episode while taking a look at what we are going to learn today. Module 1, Simple Reactions of Diosgenin. Module 2, Simple Reactions of Solasodine. Module 3, Simple Reactions of Bile Acids. Module 4, Biosynthesis of Testosterone from Cholesterol. Module 5, Biosynthesis of 17-estradiol from cholesterol. And Module 6, Biosynthesis of aldosterone from cholesterol. Module 1, Simple Reactions of Diosgenin. Let's start with simple reactions of diosgenin. Diosgenin is a steroidal sapogenin obtained from the tubers of Dioscorea species of the family Dioscoriaceae. Chemically, it is a steroidal sapogenin 3-beta-25R spirosed 5-in 3-ol and source for steroidal contraceptives, topical hormones, estrogens, progestogen, androgen and sex hormone. The sources are yams of different species of Dioscorea. Diosgenin consisting of a hydrophilic sugar moiety linked to a hydrophobic steroid A-glycan. Diosgenin is structurally similar to cholesterol and other steroids. Since its discovery, diosgenin is the single main precursor in the manufacture of synthetic steroids in the pharmaceutical industry. Over the past 2 decades, a series of preclinical and mechanistic studies have been independently conducted to understand the beneficial role of diosgenin against metabolic diseases, that is hypercholesterolemia, dyslipidemia, diabetes and obesity, inflammation and cancer. Figure 1 describes the chemical structure of diosgenin. A good naturally growing source of diosgenin in India is Dioscorea deltoidea. Diosgenin is submitted to marker degradation to obtain pregnenolone acetate. When diosgenin as the acetate is heated with acetic anhydride at 200 degrees Celsius, there occurs rupture of ring F with the introduction of 20-22 double bond to give pseudosapogenin diacetate. In a modified procedure, diosgenin acetate is refluxed with acetic anhydride pyridine in the presence of catalyst like methylamine hydrochloride. The pseudodiosgenin diacetate on chromic acid oxidation forms the keto ester diacetate, which on boiling with acetic acid gives 16-dehydropregnenolone acetate. The latter can be selectively reduced catalytically to afford pregnenolone acetate, that is 20-oxo-5-pregnen 3-beta-yl acetate. The usual name in vogue is derived from not a systematic chemical name, 5-pregnen 3-para-ol 20-one acetate. Pregnenolone derived from the ester is directly convertible to progesterone. It could be used as starting material for production of adrenocorticoid cortisone. 16-dehydropregnenolone acetate produced from diosgenin is a good starting material for preparing trihydroepinoandrosterone, that is 3-beta-hydroxy 5-androstene 1,7-one, also called prasterone, dehydroisoandrosterone or androstenolone. The oxime is prepared by reacting 16-dehydropregnenolone acetate with hydroxylamine hydrochloride and pyridine in ethanol. The oxime undergoes Beckmann rearrangement to the amide by treatment with para-acetamido benzene sulfonyl chloride in dry pyridine at room temperature. The N-amine acetamide readily hydrolyzes to the ketone dehydroepiandrosterone acetate, which on saponification gives dehydroepiandrosterone acetate, dehydroepiandrosterone. Dehydroepiandrosterone is used for the synthesis of testosterone and other androgens, ethiestrone and estradiol. This shows that diosgenin is a convenient starting material for the synthesis of male, female and male sex hormones and their modified analogues. Diosgenin is used for the commercial synthesis of cortisone, pregnenolone, progesterone and other steroid products. Now let's move on to module number 2, which is simple reactions of solasodine. Like diastenin, steroidal alkaloid solasodine isolated from Solanum species has also been used as a starting material for steroid drugs. At one time, there was good interest in the sapogenin, hecogenin, which is obtainable from plants, particularly from numerous agave species. Solasodine is obtained by the hydrolysis of primary alkaloids. Solasonine, solasodamine and solamargine is a secondary base containing one alcoholic hydroxyl group and has the composition C27H43NO2 but has only one double bond and must thus be hexacyclic. Figure 2 describes the chemical structure of solasodine. Solasodine is oxidized to an alpha, beta unsaturated ketone, and this gives on reduction a pair of epimeric alpha, beta unsaturated alcohols, one of which gives a precipitate with digitonin, but neither of which is identical with the original base. These reactions suggest a similarity in structure to solanidine, and this is further supported by the selenium dehydrogenation of the base to chimethyl 1,2-cyclopenteno-phenanthrene. Stepwise reduction of the base catalytically affords a dihydrobase solasodan 3-beta-ol analogous to dihydrosolanidine and identical with a tetrahydrobase dihydrosolasodan 3-beta-ol. Lithium aluminium hydride reduction, however, affords a different dihydro compound, dihydrosolasodine, which may be catalytically reduced to the same tetrahydrobase. Dihydrosolasodine is a stronger base than solasodine and it contains two alcoholic groups forming a OO-N-triacetyl derivative. These reactions indicate that the non-alcoholic oxygen and the nitrogen are closely associated with the solasodine, and the oxygen atom must be a part of a readily hydrogenalized ring. Oxidation of O-acetyl-solasodine leads, after hydrolysis, to pregna 5,16-diene 3-beta-ol 20-one of certain structure, and N-nitroso-solasodine with acid gives a poor yield of the sapogenin diastenin. These reactions, which define the stereochemistry of rings A, B, C and D, give no conclusive indication of the nature of the nitrogen containing portion of the molecule, but they did lead to the proposal of the structure for solasodine, though clearly the alternative is equally compatible with the evidences. The formation of 2-ethyl-5-methylpyridine, however, during the selenium dehydrogenation of solasonine, the parent of glycoside of solasodine, rules out the latter structure, and the structure for the base has finally been confirmed by partial synthesis from cryptogenin 27 to silate. Now let's move on to module number 3, which is simple reactions of bile acids. Bile acids are steroid acids found predominantly in the bile of mammals and other vertebrates. Different molecular forms of bile acids can be synthesized in the liver by different species. The bile acids are produced from metabolism of cholesterol in liver. Primary bile acids are those synthesized by the liver. Secondary bile acids result from bacterial actions in the colon. In humans, and tauroquinodeoxycholic acid and glycoquinodeoxycholic acid, which are derivatives of quinodeoxycholic acid, are the major bile salts in bile and are roughly equal in concentration. Structurally, they correspond to choline. The principal primary bile acids are cholic acids, that is 3-alpha, 7-alpha, 12-alpha trihydroxy 5-beta choline 24-oic acid, and quinodeoxycholic acid, which is 3-alpha, 7-alpha dihydroxy 5-beta choline 24-oic acid. In the liver, they are conjugated with glycine or taurine to give the glycolic acid, taurocholic acid, glycoquinodeoxycholic acid and tauroquinodeoxycholic acid before secreted in the bile. Figure number 3 describes the chemical structures of cholic acid and quinodeoxycholic acid. Deoxycholic acid and lithocholic acid are secondary bile acids and formed in the colon by bacterial deconjugation and 7-alpha dehydroxylation of cholic acid and quinodeoxycholic acid respectively. Bile acids are partially reabsorbed and conjugated. Ursodeoxycholic acid, which is 3-alpha, 7-alpha, 12-alpha dihydroxy 5-beta-choline 24-oic acid is minor acid in man. Dehydrocholic acid, which is 3,7,12-trioxo 5-beta-choline 24-oic acid, is a semi-synthetic bile acid and is prepared from cholic acid by oxidation with chromic oxide in glacial acetic acid. Quinodeoxycholic acid, which is quinodiol, and ursodeoxycholic acid, which is ursodiol, but not cholic acid, decrease cholesterol content of bile. They are used for dissolution of cholesterol-rich gallstones in patients with functioning gallbladders. Bile acids increase the output of bile and therefore called choleretic. Dehydrocholic acid is specially active in this respect and increases the volume and water content of bile acid without much change in the content of bile acids. It has been used to improve biliary drainage. It is administered orally. Sodium dehydrocholate has been given by slow intravenous injection. Best known role of bile acids are their participation in the digestion and absorption of fat. They also play an important role in several other physiological processes. These properties are related to their empipathic nature, which is due to the existence of a hydrophilic site and hydrophobic site. The hydroxyl groups oriented towards the alpha site and the carboxylic side chain afford them their hydrophilic character. The balance between hydrophobic and hydrophilic characters differs markedly among the several molecular species of bile salts. These properties have been used to predict the cholesterol solubilizing capacity of all bile salt species, but other physiochemical and biological properties of individual bile salts also may reflect their hydrophilic-hydrophobic balance. Dear friends, up till now you have gained a complete idea about simple chemical reactions of tyrosine, solasodine and bile acids. If you have missed any of the module or content, you may log on to our website www.cec.nic.in to download videos, FAQs, LOR and other contents so that you can remain updated with this chapter. Now let's move ahead to our next module, that is about biosynthesis of testosterone from cholesterol. The ability of cell to make testosterone is defined by the presence of stereogenic machinery in an appropriate cellular environment able to respond in an optimal manner to the received stimuli. Testicular androgens, that is testosterone, are synthesized in the interstitial tissue by the Leydig cells. There are a number of key points that are critical for the establishment and optimal function of steroidogenic machinery responsible for the testosterone biosynthesis in the Leydig cells. These include integrity of plasma membrane luteinizing hormone receptor signal transduction cascade responsible for sensing and responding to blood-borne luteinizing hormone, availability of sufficient amount of substrate cholesterol coming from blood or synthesized de novo storage or immediate availability of cholesterol for use in androgen production, deesterification of cholesterol, thus making available the stored cholesterol for androgen formation, integrity of the mechanism responsible for transporting cholesterol from intracellular stores into mitochondria, availability of appropriate level and combination of nuclear transcription factors controlling the expression of proteins involved in cholesterol transport and testosterone biosynthesis, maintenance of appropriate organelle structure required for optimal testosterone formation, appropriate spatial and temporal expressions of steroidogenic enzymes, mainly cytochrome P450, monooxygenases, and dehydrogenase responsible for metabolizing cholesterol to various intermediate leading to testosterone formation, and availability of the cofactor necessary for the steroidogenic enzyme action, the immediate precursor of the gonadal steroids. As for the adrenal steroids is cholesterol. The rate-limiting step, as in the adrenal, is delivery of cholesterol to the inner membrane of the mitochondria by the transport steroidogenic acute regulatory protein STAR. Once in the proper location, cholesterol is acted upon by the side chain cleavage enzyme P450 SCC. The conversion of cholesterol to pregnenolone is identical in adrenal, ovary, and testis. In the latter two tissues, however, the reaction is promoted by luteinizing hormone rather than adrenocorticotropic hormone. In the biosynthesis, first cholesterol converts to pregnenolone and pregnenolone to testosterone, which requires the action of 5 enzyme activities contained in 3 proteins. First, 3-beta hydroxysteroid dehydrogenase, that is 3-beta OHSD, and delta 5,4 isomerase. Second is 17-alpha hydroxylase and 17-20 lyase. And third, 17-beta hydroxysteroid dehydrogenase, which is 17-beta OHSD. This sequence, referred to as the progesterone or delta 4 pathway, is shown in figure number 4. Pregnenolone can also be converted to testosterone by the dehydroepiandrosterone or delta 5 pathway, which is illustrated in the figure number 4. The delta 5 route appears to be most used in human testes. The 5 enzyme activities are localized and in the microsomal fraction in rat testes, and there is a close functional association between the activities of 3-beta OHSD and delta 5,4 isomerase and between those of an 17-alpha hydroxylase and 17-20 lyase. Biosynthesis of testosterone from cholesterol is shown in figure number 4. Now let's proceed to module number 5, which is biosynthesis of 17-estradiol from cholesterol. Estrogens, 17-beta estradiol, are formed by the aromatization of androgens, that is testosterone, in a complex process that involves 3 hydroxylation steps, each of which requires oxygen and NADPH. The aromatase enzyme complex is thought to include a P450 monooxygenase. Estradiol is formed if the substrate of this enzyme complex is testosterone, whereas estrone results from the aromatization of androstenedione, which is shown in figure number 5. The cellular source of the various ovarian steroids has been difficult to unravel, but a transfer of substrates between 2 cell types is involved. These cells are the source of androstenedione and testosterone. These are converted by the aromatase enzyme in granulosa cells to estrone and estradiol, respectively. Progesterone, a precursor for all steroid hormones, is produced and secreted by the corpus luteum as an end product hormone because these cells do not contain the enzymes necessary to convert progesterone to other steroid hormones. Significant amounts of estrogens are produced by the peripheral aromatization of androgens. In human males, the peripheral aromatization of testosterone to estradiol accounts for 80% of the production of the estradiol. In females, adrenal androgens are important substrates since as much as 50% of the 17-beta estradiol produced during pregnancy comes from the aromatization of androgens. Finally, conversion of androstenedione to estrone is the major source of estrogens in postmenopausal women. Aromatase activity is present in adipose cells and also in liver, skin and other tissues. Increased activity of this enzyme may contribute to the estrogenization that characterizes such diseases as cirrhosis of the liver, hyperthyroidism, aging and obesity. Figure number 5 depicts the biosynthesis of 17-beta estradiol from cholesterol. Now let's proceed to module number 6, which is biosynthesis of aldosterone from cholesterol. Synthesis of aldosterone follows the mineralocorticoid pathway and occurs in the zona glomerulosa. Pregnenolone, which is from cholesterol, is converted to progesterone by the action of 2 smooth endoplasmic reticulum enzymes, 3-beta hydroxysteroid dehydrogenase, that is 3-beta OHSD, and delta 5,4 isomerase. Progesterone is hydroxylated at the C21 position to form 11-deoxycorticosterone, that is DOC, which is an active sodium-retaining mineralocorticoid. The next hydroxylation at C11 produces corticosterone, which has glucocorticoid activity and is a weak mineralocorticoid, that is, it has less than 5% of the potency of aldosterone. In some species, example rodents, it is the most potent glucocorticoid. C21 hydroxylation is necessary for both mineralocorticoid and glucocorticoid activity, but most steroids with a C17 hydroxyl group have more glucocorticoid and less mineralocorticoid action. In the zone glomerulosa, which does not have the smooth endoplasmic reticulum enzyme 17-alpha hydroxylase, a mitochondrial 18-hydroxylase is present. The 18-hydroxylase, which is aldosterone synthase, acts on corticosterone to form 18-hydroxycorticosterone, which is changed to aldosterone by conversion of the 18 alcohol to an aldehyde. It is depicted in the figure number 6. This unique distribution of enzymes and the special regulation of the zona glomerulosa by potassium and angiotensin II have led some investigators to suggest that in addition to the adrenal being 2 glands, the adrenal cortex is actually 2 separate organs. Figure number 6 depicts the biosynthesis of aldosterone from cholesterol. With this, now we come to the summary. Diosgenin is steroidal sapogenin obtained from the tubers of Dioscorea species of the family Dioscoriaceae. Chemically, it is a steroidal sapogenin, that is 3-alpha 25R spirosed 5-ene-3-ol. Diosgenin is a convenient starting material for the synthesis of male, female and male sex hormones and their modified analogues. Diosgenin is used for the commercial synthesis of cortisone, pregnenolone, progesterone and other steroid products. Steroidal alkaloid solosodine isolated from Solanum species has also been used as starting material for steroid drugs. The bile acids are produced from metabolism of cholesterol in liver. Many hormones are made from cholesterol. All mammalian steroid hormones are formed from cholesterol via pregnenolone through a series of reactions that occur in either the mitochondria or endoplasmic reticulum of the adrenal cell. With all these informations, here we come to end of today's lecture. Do keep in mind what we discussed today. It is time for you to self-study. This is Ankita Tiwari signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQ, quiz, LORs. The website is www.cec.nic.in. Till then, keep studying. Goodbye. Thank you.